to the NPTEL lecture series on animal physiology. So, we finished with the gastrointestinal physiology and now we will be moving on to metabolism and thermoregulation. So, this is section 13 and there are dedicated 2 lectures for that. So, what we will essentially do in this section? So, as of now we studied when we take the food, we chew it up. The first set of reactions start by the salivary gland, then the food passes through the esophagus, kind of a tunnel, and goes to the stomach in an acidic environment. There it is exposed to hydrochloric acid, and from there it moves on to small intestine. There is significant amount of absorption takes place, and then to the large intestine likewise and gets rejected. During this process, we have appreciated how pancreas secretes different exocrine secretion and endocrine secretion of insulin and glucagon. We talked about the secretion from the liver, the bile juices, how they help in emulsification of the fats and everything. So, as of now we talked about in terms of the anatomical features and the movement of food through this anatomical structure. What we will essentially do now is we will give it a biochemical flair. So, what does that mean is, so food basically consists of 5 different components. So, one of the major component is water, carbohydrates, lipids, proteins, some minerals which eventually form the electrolyte in our body, and some vitamins and some hormones which are needed in very, very low concentration, and a series of minerals. Okay. So, these are absorbed. First of all, these are degraded in through series of reactions. So, what we will do, first of all, we will talk about or we will classify the different enzymes which are involved in breaking down of carbohydrate, which is one of the major component of food, the lipids and the proteins. Once we talk about those, then we will talk about 2 or 3 different cycles by which these enzymes are involved in 2 distinct processes of metabolism. So, metabolism, this word could be could be divided into 2 parts, or the addition of 2 words could be considered in metabolism. So, one is catabolism, the other one is anabolism. Catabolism is a process when there is a breakage of a bigger molecule into smaller pieces through enzymatic action, whereas the reverse process, when the smaller molecules added up to make a bigger molecule, say for example, small peptides making a polypeptide chain, or small single carbohydrate sugar molecules making a starch molecule or a glycogen molecule. That is a constructive process, or where that involves the integration of the smaller molecules, and that process is called anabolism. And both anabolism and catabolism are regulated by a series of enzymes. So, start off with what I will do, I will enumerate the different enzymes which plays significant role in our in the digestion of the 3 different chemical components. So, coming back to the slides. So, we are essentially into section 13, metabolism and temperature regulation. This is the second part of this. So, today we will be dealing with metabolism, which is our lecture 1. So, coming to the different components of the food we talked about, try to divide food in terms of chemicals, chemical origins. So, then will be carb, lipids and proteins. And if I classify the different enzymes which are involved in it, for the carbohydrates you have maltase taking care of maltose sugar, sucrase breaking down the sucrose sugar, lactase breaking the milk sugar or the lactose. Then you have secretion from pancreas, pancreatic amylase or alpha amylase. You have salivary amylase taking place from the saliva. Okay. Then from there, let us move on to the enzymes which are involved in breakage of proteins. So, most of them are called proteases. So, proteases includes carboxypeptidase, you have chymotrypsin, you have dipeptidase, peptidase, so breaking down the peptide bond, dipeptidase, you have enterokinase, elastase, then you have pepsin, then you have renin, then you have trypsin. These are some of the major ones, and then coming to lipids, they are mostly falling under the title of lipases, which includes lingual lipase, and you have pancreatic lipase. And then you have in that section a smaller section you can add, which are called nucleases, which includes mostly pancreatic nucleases. So, coming back, so I have enumerated the list of different lipases, different proteases, different carbohydrate breaking enzymes, and the nucleases. So, these are secreted by different parts of the digestive system or different part of the body. So, what I will do now, I will highlight which part of the digestive system is. Some of them are self-explanatory. It's a pancreatic lipase, means it is secreted by the pancreas, but there are others which are secreted by multiple organs of the digestive tract. So, we will just enumerate them. So, we will put them in different color, so that makes more sense. Okay. So, coming to the maltases. So, maltases are secreted by small intestine. I am just putting SI, means small intestine, sucrase small intestine, again lactase small intestine, okay. Pancreatic lipase, as it says, that is by the pancreas, salivary amylase, it is by the saliva. Among the lipases, lingual lipase. So, lingual lipase is basically the glands of tongue. So, basically all the way in the mouth, GUN, one second, GUE. Then you have pancreatic lipases, which is secreted by the pancreas, nucleases by the pancreas, and among the proteases, so carboxypeptidases by the pancreas, then you have chymotrypsin by the pancreas, and then you have dipeptidases, that is by the small intestine, then you have enterokinase by the brush border and the lumen of the small intestine, SI. Then you have elastases, are secreted by the pancreas, dipeptidases by the small intestine, then you have the renin, which is secreted by the stomach, then you have trypsin, which is again by the pancreas, and then you have the pepsin, which is secreted by the chief cells of the stomach. So, and yeah, so coming back. So, if you look at it, so all these different enzymes are secreted by the different parts of the digestive system, and in that whole process, there is a division of labor. So, once it goes to the mouth, it is chewed up by the teeth, and these couple of enzymes are secreted either from the salivary gland or from along the mouth. During in the oral cavity, there are the secretion, and the food moves. Then there are renin and few other stomach enzymes are secreted from there in the intestine. But if you see the huge chunk of this concentration of these enzymes are concentrated along this in the small intestine and around the pancreas. So, this is the zone where most of them are concentrated, okay, along the small intestine and the pancreas. Okay. So, from here what we will do, after giving you this overview of these different enzymes which are getting secreted, we will move on to the different catabolism processes and the anabolism processes which are involved in the whole process. So, before we do so, I will just take one step back. So, I talked about all this and all these different kind of enzymes in the slide. We will devote one more slide talking about all the different products of carbohydrates, fats and proteins, how their smaller components are getting absorbed and which part of the digestive system they are getting absorbed. Once we do that, then we will move on to the catabolism and anabolism, the different cycles which are involved, the Krebs cycle, glycolysis, gluconeogenesis and all those processes which are regulating the different aspects of metabolism within our body. Okay. So, coming back to the slide now. Okay. So, what we will do essentially at this point, we will talk about who is absorbed where. So, we talked about all the components. Okay. So, the carb, we have lipids, and we have the proteins. So, what is happening in the oral cavity? In the oral cavity, here you have the salivary amylase. Okay. So, from here it is getting broken down. So, let us write down on the other side, esophagus, then you have the stomach. I am just putting the initials, okay, small intestine, and then you have intestinal mucosa, and then you have the blood stream where the major absorption is taking place. Okay. So, and during this course of movement of the food, what is getting affected where? So, in the oral cavity, it is basically salivary amylase. From here, it moves all the way to the small intestine. In the small intestine, it is basically one second. I am just putting it slightly wrong. Okay, let me put some. Okay, let me put lines that way. It will be easy to break it down. What is happening where? Fine. Now it's fine. So the carbohydrate is moving all the way to small intestine after getting broken down into by the action of salivary amylase. So here you have the pancreatic amylase which comes into play, pancreatic alpha amylase, and this pancreatic alpha amylase break down it into disaccharides and monosaccharides, and from here these are absorbed by the intestinal mucosa. So these are the intestinal mucosa where it is getting absorbed. So basically these different component monosaccharides and disaccharides through facilitated diffusion and co-transport, okay, diffusion and co-transport, and to further diffusion moves into the capillaries. They are all in the form of monosaccharides. So this is how the carbohydrate is being absorbed. So, initially you chew the food, salivary amylase acts on it, then the food is kind of partially broken down, the carbohydrate part, then those partially broken down carbohydrates moved on to the small intestine. In the small intestine, it receives secretion from the pancreas. So, the pancreatic alpha amylase helps in breaking it down into the monosaccharide and the disaccharides, and then these disaccharides and monosaccharides are absorbed by the diffusion or facilitated diffusion or co-transport into the mucosa, intestinal mucosa. From intestinal mucosa, they are transported to the bloodstream. Okay, so this is the fate of the carbohydrates. So let's talk about the fate of the lipids. What is happening to the lipid molecules? Okay, so lipid molecules out here are under the action of lingual lipase. We talked about this. So from lingual lipase, then it moves into the small intestine, where the bile salts play a key role in emulsifying bile salt and pancreatic lipase. These two play very critical role forming monoglyceride fatty acids in micelles. Okay. Monoglyceride fatty acid micelle. So, these micelles then diffused inside the intestinal mucosa and they form triglyceride, then they form chylomicron, and these chylomicrons are then moved via exocytosis. So, here basically exocytosis is taking place and they move on to the bloodstream, bloodstream and the lacteal glands. Okay. So, this is essentially is the fate of the carbohydrate, which is fate of the lipids, which is moving through the blood from the mouth all the way to the intestine, where by the action of the bile acids are getting emulsified and broken down into the glyceride, monoglyceride, and then it form triglycerides. From the triglycerides, it through the chylomicrons and everything, it is getting exocytosed, and then it moves into the blood vessels and travel through the specific organs wherever it has to do its function. So, from here we will talk about the fate of the carbohydrates, okay, which we haven't talked yet. So, the proteins. Okay. So, the proteins, once their first action takes place in the stomach, where basically your pepsin comes into play. So, proteins become polypeptide, next action. From here they move on to the small intestine, where there are series of enzymes which are involved: trypsin, chymotrypsin, elastase, carboxypeptidase, carboxypeptidase. Okay. So, that form the short amino acids. From there it diffuses into the intestinal mucosa. In the intestinal mucosa, so mostly this is facilitated diffusion and co-transport, okay, just like the carbohydrate, facilitated diffusion and co-transport. So, here there are two different enzymes comes into play, exopeptidases and dipeptidases, okay, and from here it moves on to the blood vessels. So, essentially in that whole process, what is happening is the huge protein molecule is getting broken down into smaller amino acids. These amino acids through diffusion and co-transport processes are absorbed. So, one absorption which I haven't talked as of now while talking about carbohydrate, lipids and fats is how the different ions are getting absorbed. What are the different processes? So, what we will do, I will talk about the different ionic electrolytes. I will just give you an enumeration of, I will enumerate the list of the different ions which are involved and how they are transported across the body. and from there we will move on to the catabolism and the anabolism process. So, let us enumerate the different ions which are involved in it in our body. So, essentially we have sodium, calcium, like I am only talking about the major ones, potassium, magnesium, iron, chloride, iodine, HCO3 minus, NO3 minus, PO4 3 minus, SO4 2 minus, and you have certain, after this list you have water soluble vitamins and fat soluble vitamins. So, which includes A, D, E and K, B and C. Okay. So, most of these are absorbed through the intestinal mucosa by process of either active transport. So, mostly they will be either active transport or diffusion, and sometime carrier mediated diffusion, and they are also absorbed not only in the intestinal mucosa, these are also absorbed along the kidney. As we have discussed in the kidney, they are along those along the nephron, there are different zones where some of these are getting retained by the body, and that is what helps in the electrolyte regulation within our body. So, from here what we will do, we will move on to the overall metabolic, like overall layout of the catabolism and anabolism to give you an idea. So, we have talked about the enzymes, we have talked about where these enzymes along the digestive tract are involved in the breaking down of the food components. We talked about, have enumerated the different ions and the vitamins which are being used by the body for different metabolic processes, and how they are transported across the lumen into different parts of the body where they will be needed for further anabolic action or catabolic action. So, now we will move on to the overall layout of the catabolism and the anabolism process. Okay. So, let us getting back to the overall classification of anabolism and catabolism. So, basically, this falls under the A of cellular metabolism. So, cellular metabolism could be divided like this. You have these organic molecules, which includes your amino acids, carb and lipids. Okay. These organic molecules either via, as we have seen, diffusion or transport, they are all these two major modes, they entered into the nutrient pool of the cell. Okay. Here there are two possibilities involved in anabolism. So, where basically you are synthesizing, so if this is the part of the cell, okay, synthesizing new molecules, or they are involved in catabolism. So, what is essentially happening in catabolism? They are involved in different kind of breakage processes, and which generates heat. And similarly, during these catabolizing processes, ATP is produced. This ATP helps in the synthesis of different kind of these anabolic processes, as well as other aspects like movement, cellular movement in terms of mitosis, when there is a movement of the different kind of cell organelles and the chromosomes which are moving, the transport, because transport needs enormous amount of ATP molecules, co-transport, ATP dependent transport, movement of the muscle. There are several zones where that ATP which is produced as a part of, let's coming back to the slide, the ATP which is produced as part of the catabolism process is being used. Okay. So, this is overall geometry if I had to tell you that how all these things are happening. So, this is how this whole process is running. And talking about the metabolic turnover and cellular ATP production. So, metabolic TO, I am just putting a short form of the turnover and cellular ATP production. Okay. So, essentially what is happening is that during, so I am representing anabolism and A. The anabolism, there are two possibilities, either these anabolic products are secreted by the cell, if this is the cell, okay, or they are stored, or these anabolic products have multiple roles. So, these could be broken down and helps in the different physiological function. Of course, for that you need the energy rich molecules ATPs, which are of course produced by the mitochondria, okay. And this anabolic products are helping growth and maintenance. And these growth and maintenance eventually leads to a lot of catabolism processes which enhance the nutrient pool of the body, and this nutrient pool is again used up by the growth and maintenance. Okay. And there is continuous supply in the form of food, and which are in the form of amino acids, triglycerides and sugar, which are coming into play. So, and if you look at the nutrient used in cellular metabolism, cellular metabolism. So, it is something like this. So, you have the mitochondria. So, where the TCA cycle is taking place, we'll come to that, which is producing carbon dioxide, and in that process out here you have the ATP production and using electron transport chain, and you have these small carbons like from glucose, amino acids, which are all involved here, fatty acids, okay, and you have this triglyceride, glucose are coming from glycogen, and then you have the amino acids coming from the proteins. So, this is your essentially the nutrient pool of the cell, and this is essentially the storage pool. So, whenever our individual cell always have two phases of storing. One will be the nutrient pool, which is readily available, like they are broken down in small pieces, they are readily available for energy production, and there is another pool which is the storage pool, where you have the bigger molecules which are sitting there. So, those bigger molecules, when they are breaking down into a smaller nutrient pool, they follow the catabolic route, okay. And then again those molecules are being utilized for energy production, and simultaneously in order to have those storage molecules, you have to go through the whole anabolism process, okay. So, this is the overall kind of layout of the metabolic events at the cellular level. From here we will move on to the three specific zones. We will just browse through it about carbohydrate metabolism, lipid metabolism and protein metabolism. Okay. So, let us moving on to the carbohydrate metabolism now. Okay. Carbohydrate metabolism. Okay. So, carbohydrate metabolism, to get the basic reaction is glucose plus oxygen leading to carbon dioxide and water formation. So, this is your glucose and here you have the water. So, this is the basic most fundamental reaction of carbohydrate metabolism. So, the carbohydrate metabolism is or the formation of glucose molecule in this whole process and utilization of the glucose molecule. So, these are the most I should say most fundamental energetic bioenergetic processes in our body. So, in this section we will talk about the glycolysis, Krebs cycle and how all these processes are intertwined in the gluconeogenesis processes and in the building up processes. So, coming back to the glycolysis process, it is happening. So, the first the word which comes here will be glycolysis. So, whenever you see any word called lysis means breaking it down into. So, basically what is happening is that glucose is broken down into pyruvic acid. This is basically what we meant by glycolysis process and what it requires. There are certain basic requirements. The first one is glucose molecules, first one. The second one is cytoplasmic enzyme. Third one is ATP and ADP. Fourth one, you need inorganic phosphate, and I need NAD and NADH. So, nicotinamide adenine dinucleotide. So, NAD stands for nicotinamide adenine dinucleotide. Okay. So, these are the 5 major things what is needed for the glycolysis process. So, what are the steps in glycolysis? So, as we already talked about, so this is happening inside the cell. Okay. So, let me demarcate it like this. So, this is the green line what you see is inside the cell. So, the glucose is coming inside the cell. So, here is you have the glucose from glucose is converted into glucose glucose 6 phosphate, and that in that process there is an ATP to ADP conversion. So, that needs energy, ATP forming ATP, and there is a lot of energy which is needed in that process. From glucose 6 phosphate it forms fructose 1,6 bisphosphate. This step is also energy requirement processes, ATP to ADP, adenosine triphosphate to adenosine triphosphate. At this stage there are 2 fates, possibly 2 fates. One is glyceraldehyde 3-phosphate, or it could form dihydroxy acetone phosphate. Okay. So, dihydroxy acetone phosphate can again transform into glyceraldehyde 3-phosphate, and from here this aldehyde 3-phosphate forms 1,3 bis phosphoglyceric acid. And from there, and at this stage from 1,3 bis phosphoglyceric acid to phosphoglyceric acid, there are 2 ADP molecules transformed into 2 ATP molecules. For the first time, the reverse process is taking place in this cycle, and from here it gets rid of water molecules, and then it forms something called PEP, phosphoenol pyruvate. Phosphoenol pyruvate. Okay. And from phosphoenol pyruvate, it forms pyruvic acid. And this, during this formation, again 2 ADP molecule is utilized to make 2 ATP molecules. So, if you look at this process at these 2 zones, out here and out here, you have developed ATP molecules, whereas here you have consumed, this is basically you have broken down ATP molecule, and here you have made ATP molecule. And if you look at the energy summary in terms of the ATP, so there are -2 ATP, and here you have +4 ATP, and essential gain is +4 -2 is +2 ATP in this whole process. And here it is, I will highlight one thing. So, you saw this reaction, I drew it so easily, but think of it, anybody can draw that reaction, but how many enzymes are involved in it? So, at every step of this reaction, from glucose to glucose 6-phosphate, bisphosphate, phosphoenol pyruvate, pyruvic acid, all these are regulated by enzymes. So, as of now, we only talked about the enzymes which are involved in breaking down of the carbohydrate, amylase, alpha amylase, pancreatic alpha amylase, salivary alpha amylase, lactase, galactase, so on and so forth. But they are just a big chunk of carbohydrate which you are breaking down into, tweaking into small pieces. But then that individual glucose molecule is going through from glucose to pyruvic acid, what I just now drew for you. You must have observed that how many reactions are involved in that, and each one of these reactions are regulated by a specific enzyme. Without that, this process won't happen. So, that is the reason that the cellular metabolism needs all those different enzymes at one point of time, other than the enzymes what we have already enumerated. And we have seen this whole glucose breakdown process to pyruvic acid finally lead to the formation of 2 energy rich molecule. So, every molecule of glucose at the end produce 2 energy rich ATP molecule. That is essentially is the take home message from this process, what is happening in the breaking down process of glucose. It is a catabolic process, by the way, and this catabolic process where glucose is further. So, first series of catabolic processes like this. So, if I had to put, so you have this huge carbohydrate molecule, okay, huge carbohydrate, then this forms a small, say, glucose, okay, small molecule. So, this is the catabolisms. I am just representing with C, the pink C. Okay. So, there is a catabolism process. From here, it forms pyruvic acid. So, this is another catabolism processes. Okay. And during this whole catabolism processes, it leads to formation of ATP. So, each one of these molecules lead to the formation of an ATP molecule, 2 ATP molecule. So, this is very essential, and just once they are forming pyruvic acid, it is not end of the story. That pyruvic acid went inside the mitochondria and leads to a cycle called TCA cycle, tricarboxylic acid cycle. The tricarboxylic acid cycle is fed into the electron transport chain of the mitochondria, and there it plays another set of reactions for the anabolic and the catabolic processes. So, what we will do today, we will go through the tricarboxylic acid cycle of that pyruvic acid which has formed. Okay. Coming back to the one second, let me just go through this one. Fine. Okay. So, now you have the pyruvic acid out here, right? Pyruvic acid. This pyruvic acid through coenzyme A and process utilizing an NADH out here become acetyl CoA. This acetyl CoA form citric acid, then it form a 5-carbon structure, and then it form a 4-carbon structure. Then that 4-carbon structure in that process eventually become acetyl coenzyme, and this is all happening out here inside the mitochondria. Okay. And out here what is essentially happening during this process is that there are this, keep on looking, I am just, so this is generating ATP out here. There is always a proton gradient which is getting created in this process, and citric acid 6 carbon. So, this is basically 6 carbon gives away a CO2 out here, and 5 carbon it is giving out a CO2 out here, and these proton gradients which are getting generated out here, 2H, 2H, okay. These are different proton gradients. So, all these proton gradients along here are being picked up by this different FADH2, NADH. They are, so where you have the other, okay, out here through NADH. So, here also NADH, and all these become part of the electron transport chain of the mitochondria. So, they are involved in the electron transport chain, and this is one of the most key reaction TCA cycle which is called. So, this is one of the most key reaction of the body which is involved in feeding the electron transport chain and formation of the ATP molecule. So, what is happening, what is the fate of these different NAD and NADH system is inside the mitochondria is, one second, okay. So, these NADs through a series of reactions. So, basically this whole electron transport chain is going is at different gradient going through CO2, cytochrome oxidase, cytochrome B, cytochrome C, cytochrome A, cytochrome A3. Likewise, all these different reaction processes and different donors of the electron along this whole process inside the mitochondria is leading to the formation of final end product of this whole game is ATP production. So some of these things which I haven't already discussed. So, this is FMN is basically flavin mononucleotide. Okay. And these are the coenzyme A, coenzyme Q, coenzyme A, and different other factors which are involved in this whole process. So, if you look at it, so we started outside the cell when the glucose moieties started coming in, the glucose moieties got transformed, and from that point onward through the formation of glucose to pyruvic acid, and then this pyruvic acid gets inside the mitochondria, and through acetyl coenzyme A formation and everything leads to a proton gradient, and this proton gradient is eventually involved in the production of the ATP molecules out there. Okay. And this proton gradient, which is created along the mitochondrial membrane, is one of the critical aspect of bioenergetics, which is kind of the lifeline. That some of these reactions are the bioenergetics basis of the life form on the floor of Earth. So, in that process, what we will do now, we will summarize the whole process in terms of the ATP production before we move on to the lipid and the protein metabolism. So, talking about the summarizing this whole process in terms of the lipid production, in terms of the ATP production. So, this is what is essentially happening. Okay. So, during glycolysis, you have 4 ATP, then minus 2 ATP is being used, and what you get is 2 ATP. Okay. Now, at the electron transport chain, now we are entering into the electron transport chain. So, you have 4 ATP, then you have 24 ATP in the TCA cycle, and you have another 4 ATP which is formed different stages of the cycle, another 2 ATP which is formed. And sum total of the cell during this whole net gain of the cell from aerobic catabolism from one glucose molecule, what we essentially get is 36 ATP molecule. And how that happens, we are going to come now. I will draw the chart to show you how this whole thing is happening. So, you have glucose and you have 2 ATP forming 2 ADP. So, you are utilizing ATPs now, okay? And you have 4 ADP forming 4 ATP. So, this is the first set of reaction which leads to plus 2 ATP formation from glucose 6 carbon to pyruvic acid. Now, pyruvic acid is entering inside the mitochondria. Okay. Inside the mitochondria, pyruvic acid goes through to form acetyl CoA, and acetyl CoA enter inside the TCA cycle. And during that process, it forms from pyruvic acid to acetyl coenzyme A, it is forming NADH. These NADH through the electron transport chain. So, there are 2 NADH here, and there are 6 NADH, and there are 2 FADH2, which are all outcome of the tricarboxylic acid cycle. So, this and via, and there are few, there is one more source by which it comes. So, during formation of glucose to pyruvic acid, there is another, I will just show it to you in another color. So, that there is an NAD molecule which is formed, which is fed from here, from outside. These NAD molecules also become part of the electron transport chain. So, if you look at it, so there are 2 NADH molecule which are coming in from here. There is another 2 which is coming from here. So, let me highlight them. So, 2 coming from here, 2 coming from here, 2 plus 2 4, and you have 6 NADs coming from fed from here, 2 FADH2 fed from here. And in that process, what is happening here? So, this is leading to the formation of 4 ATP out here. 2 NAD is again leading to the formation of 4, 2. So, 2 is leading to the 4, this 2 is leading to another 4, this 6 is leading to 12, okay. And you have 2 FADH2 molecule which is leading to formation of 4. So, if you add up this 4, 12 plus 4, 16, 4, 4, 8, 8, 4, 12, 12, 12, 24, then you are making 26. Okay. So, essentially what is happening is that you have 2 NADH, 6 NADH, 2 FADH2, okay, 2 FADH2, and so 2 NADH and 6 NADH is leading to the formation of actually here multiplied by 2. So, basically out here, just let me out here you are making 24 ATP molecules, and there is 2 more which is coming from a GTP and GTP, and this eventually leads to 4 ATP here, 24 ATP here, you have 4 ATP here, and another 2 coming from the tricarboxylic acid cycle, and what you essentially get is that 2 plus 4 plus 24 plus 4 plus 2, 4 plus 2, 6, 6, 10, 10, 14, 14, 16, plus 3, 36. So, this is basically what is happening in the process of ATP production. There are 36 ATP molecules which are generated from a single glucose molecule when it goes through the whole process of pyruvic acid formation and through the tricarboxylic acid cycle. So, what we will do next is that in the next class, I will close in here. In the next class, we will move on to in the gluconeogenesis process, lipid metabolism, and protein metabolism, and thermal regulation. Okay. Thanks a lot.